My name is Jan Singh. On behalf of Education Committee, I'm moderating this coffee hour focused on establishing your lab as a new PI. To lead this coffee hour discussion, today we are graciously joined by two esteemed speakers, Dr. Taha from Vanderbilt University Medical Center and Dr. Jinkyung Kim from Washington University School of Medicine at St. Louis. Dr. Kim's research has focused on developing in vivo imaging techniques and applying them especially to the auditory periphery organs like cochlea. During her graduate work, she developed and optimized synchrotron X-ray tomography system and performed quantitative 3D microstructural imaging of the organs, which laid the foundation for her postdoctoral research in auditory system at Stanford University. There she used in vivo optical coherence tomography to show that osmotic stabilization prevents cochlear synaptopathy after blastoma. The OCT lacked cellular level resolution, and to address this, she developed a surgical method enabling in vivo real-time visualization of cochlear cells in hearing mice. This technique allowed her to track ototoxic drug transport into the cochlea and uncover a novel uptake pathway. She further expanded its utility by delivering genes, drugs, or sensors through the semicircular canal without damaging hearing or balance function. Dr. Kim has now brought this in vivo cochlear imaging platform to her independent lab at Washington University in St. Louis, where she's applying this platform to explore cochlear physiology and pathology in vivo, aiming to uncover mechanism of hearing loss and developing strategies for its prevention. Thank you, Dr. Kim, for joining us today. And another speaker who joined us is Dr. Taha. Dr. Taha is a surgeon scientist and investigator with a primary appointment in otolaryngology, head and neck surgery, and secondary appointment in cell and developmental biology and hearing and speech sciences. His research group aims to identify molecular pathways that can be manipulated to stimulate hair cell regeneration in mammals following injury, with the ultimate goal of restoring hearing and balance. The team pursues two main approaches. Firstly, addressing fundamental questions of development and regeneration, and conducting translational research using human inner ear tissues. By leveraging cutting-edge computational tools alongside in vivo and ex vivo experiments in mouse and humans, Dr. Jan's team seeks to decipher mechanisms of mammalian hair cell regeneration. On the clinical side, Dr. Jan cares for patients with hearing loss, balance disorders, and lateral skull-base pathology in both the clinic and the operating room. He also serves as a site investigator for sponsor-initiated first in-human inner ear gene therapy clinical trials targeting otophilin-related hearing loss and anti-VEGF for vestibular schwannomas. Dr. Jan, thank you for joining us today for this coffee hour. So, as we know, establishing your own lab as a new PI marks a significant academic milestone. Although this career breakthrough comes with exciting opportunities to establish yourself in the field, but also there are new responsibilities and challenges that come alongside this new journey. Today, we would like to learn from both of you about your initial experiences and how was the journey of establishing your own labs go as a new PI. Before we go forward with the discussion, I would like to encourage our audience to put their questions in the chat box, and we will take their questions alongside this discussion. Dr. Kim and Dr. Chan, I will let you lead the discussion now. Either one of you can go first. Thank you.
Thank you, Janelle. Why don't you go first, Taha?
You can go first. Thank you so much for that very, very kind introduction. Go ahead, Jinkyung.
Oh yeah, thank you. And hi everyone, nice to meet you. I'm Jinkyung Kim. I'm at WashU now. Then it's been about 2 years just I opened the lab. And then I'm still, very struggling, but I learned from great senior PIs and then also got a lot of advice, when I opened the lab and still now as well. So I'm more than happy to share, several tips like when, you open your own lab. And I just want to share probably three things, but like always welcome to ask any question if you have, So I think first one I just want to share is probably um, anyway, you're gonna sign the offer probably before several months, sometimes a year prior to you like physically join the new department, right? And I just say you can order the items, after you join, but the problem is that if your lab required a big like equipment or some expensive one, you need to negotiate with multiple vendors. And then, even you just place the order, you need some help from just administrations. And also like even after that, shipping takes several months. So I think, in my opinion, the first thing you can save your time after join new department because your tenure clock, would start when you physically there, right? But like probably the moment you just, you negotiate your offer letter, ask your like chair to like allow you to spend money before joining that day. I'm sure that like probably most of the chairs though doesn't have any issue with it. So the main equipment I currently have is two photon in vivo system. And then basically it took about half year to get, but like since I just ordered it, negotiated like before half year before I joined WashU, and that saved my time half year. And then basically I can align all the laser or set it up everything there. I mean, when I just joined here. And the second thing, another part you need to do after you join, I think you should promote yourself within the institution. Definitely, people in your department would know, but like there's a lot of collaboration chance also, especially if you are a PI, but I mean PhD, but not in the just um, basic science department. Like for example, you are in medical school, you may need to like promote yourself to like other graduate student or PIs in other department because that provide a potential graduate student to you, and plus so like great collaboration opportunities. So if you just ask can I have a like give a talk to other different programs in the institution, also like probably the another institution, they are welcome to make some spot for you. And then that really helped. And then last thing, I think you need to find right people, your people. And I know this is really hard, and a lot of senior PI says that it's kind of luck, to meet the right person and then, good person. But like at least I think you can increase the like probably the potential like or possibility to have like good or and also right person. So for example, try to advertise your lab like multiple way. I just put the hiring advertisement like a lot, like job archive, also some conference website. And also I use the conference and then you try to find, some graduate student, present like the research which is closely related to mine. I just, stop by their poster and then introduce yourself. And actually that helped a lot. And another thing is that like, you are gonna get probably a lot of like applications, but if you like one person, I usually, like call directly to their reference instead of getting the letter. And that helped a lot because my current postdoc had a lot of red flag, but I really liked him. And then ends up I just think he's amazing. And then he's doing super great. But the reason why I can hire him because I talked with his like multiple references, like probably like several hours meetings, and then like got better sense about him. So like having great and the right person will change a lot to you, because if you can, probably if you pick wrong person, like you may just, find another one, but it's your time, because as a new PI, we have very limited money and time. So you are gonna probably only have a couple of trainee when you start your lab, but one of them is not good. It's you lose your time, and then of course, like, money, and then probably chances. So I think, you put a lot of time and effort to find right people, and that's probably the tips I can share here. Yeah, Taha, why don't you
Thank,
share your tips?
thank you, Jinkyung. That was great, and I see a lot of familiar names on the in the audience, including some clinician scientists as well. So just to kind of dovetail based off of what you had already mentioned, few tips, for there's not really as much of like an onboarding process when you start a new job. You meet with somebody in your department, whether that's otolaryngology or the basic sciences, and they kind of tell you, well, here's your benefits, here's, well, you get your paycheck and whatnot, and then you're just kind of left on your own. You show up and your lab may or may not have anything in there. So you kind of have to take things into your own hands, and it's good to get to know people, particularly in your department. I'll get back to that in a second, but going off of what was already mentioned is getting the onboarding process and get an email as soon as possible. A lot of places will let you start an animal protocol before you show up. There's a thing called a like new investigator protocol or no procedures protocol or whatnot, but a lot of times you can actually set up an animal protocol before you show up, and that allows you to order mice or transfer your animals or whatnot and get that process going, because that can take a few months and that'll save you a lot of time. So as soon as you get an email address after you signed your contract and whatnot, then you'll at least be in the system and you can start the protocol. You can ask for neighbors to get their protocol or your current lab, whether you're working with mice, monkeys, or whatever, zebrafish or whatnot. So that's really important. Likewise, for those of you working with human specimens, you can get IRBs going, and again, those take on the order of months usually, and the sooner you get the ball rolling, the easier it'll be when you show up physically, because as soon as you show up physically, you'll have additional responsibilities. And then for those of you who are clinician scientists, getting your licensing and credentialing going ahead of time, really, really important. And then you can also do a lot of all of the online safety trainings and these things. Like you can't put in the mouse protocol until you do all of the safety training and these things. So you can get a lot of those things done ahead of time because it will just save you time. Going off of again what Jinkyung already mentioned was that learn the hiring process, and it's different everywhere that I've been. And some places require you to have a open position on their website up for 3 weeks. Other places it's for 6 weeks. So whatever it is, learn the hiring process. In general, there's one person in your department that has the title of something like, department manager or hiring manager, or some sort of title like that. Whether you're in a clinical department or a basic science department, it's really important to get to know who that person is. And that person usually is your liaison with the entire institution's HR department, which is going to be different from your department. So there are certain rules that kind of apply across the board, and they're not going to change the rules for you. So you have to kind of conform yourself to whatever rules they have. For example, so my first job was at UCSF. I was there for 2 years before moving to Vanderbilt just over 3 years ago. And they had a rule that, that you had to have an open position for at least 6 weeks before you could hire somebody, or at least that's what I was told. And so what I learned to do was keep sort of an open position almost all the time, and it would expire after 18 months. And so they're like, oh yeah, everybody does this. And so that way even if you have somebody identified, it just makes the hiring process a lot easier. I want to extra emphasize what Jinkyung always mentioned: get on the phone and talk to references. People will not write down everything in a letter, but they will talk to you on the phone to tell you if there are any red flags or not. And as was already mentioned, those first couple of hires are really important. It's very different if you have a large lab with like 20 people in it and you hire somebody that doesn't work out, right? They can probably absorb that and there's a lot of resources. It's very different when it's like you and one other person versus you and two other people. So very important to do your homework there. The other thing I would encourage you to do is build a lab website. It's pretty easy to do. I have mine on like on Google websites. I pay $20 a year for the a URL name, the site. And so build a website because people will not know you, but they'll look you up, like that graduate student or somebody you bumped into. They'll just Google your name, and if you have a nice website, they're more likely to shoot you an email versus, they just can't find you. Takes like a few months to get onto the departmental website and whatnot. So it depends on where you are, but I would say, get a lab website going, and also social media. I mean, we have a social media, we have an X Twitter now, X account. I rarely use it, but it's still sort of claiming your domain if you have time. I don't have much time to deal with that these days, but at least the lab website I try to keep up. Um, other opportunities to kind of find people, as Jinkyung already mentioned, posting on different websites. I certainly used also, you use ARO, but you can also use other other areas that are outside of the field. So we do some developmental biology. So I put an ad in, selling, in developmental biology conferences and meetings. Sometimes you have to pay for them. It's like several $100s, but you can, charge that to your account. And then you can use LinkedIn. You can use the HR department has their own thing. So I would cast a wide net. It's unlikely that you'll find somebody who's coming in and already has a PhD in hearing biology. If you do, that's awesome. But just because somebody's outside the field doesn't mean that you can't teach them and they can do good science and be good people. Um, and then as far as other things go, is find a scientific home early. Start attending, whether it's like weekly seminars or every other week. So at Vanderbilt here, we didn't have, up until recently, we didn't have like another inner ear biologist. So my first meetings, we're part of the Epithelial Biology Center. The inner ear is epithelium, so I'm part of that group, and so my people also go to those meetings. So when you have 2 or 3 people in lab meeting, it's different than when you have other folks in there and can bounce ideas off of each other. So even if it's outside of your field, it's okay. There's going to be good scientists there who are smart and doing science at a high level. You want to associate with them and go to their meetings and participate and present your ideas. Even if it's, ear related, there will still be very smart people in the rooms. And I always try to go to a room where there's smarter people than me, so that way I can learn stuff from them. And it also frees up a little bit of your time if you have your, whether postdoc or research assistant or graduate student, going to some of these meetings and learning on their own and creating a little bit of community. And that's really important. Like maybe the day you're working on a grant or you're in clinic or something, they have a question about a Western blot or about a PCR or something, they can go to the neighbor and say, hey, what do you think about this kind of thing? So that's something else that I would encourage doing. Other things for finding people to work in your lab: if you can, get on the committee for the PhD admissions folks, because you'll start interviewing people. I've done that the last 2 or 3 years, and I've started to get, folks in the interdisciplinary graduate program actually want to rotate in my lab, because we're not like, again, a lab with like 20 people who've been established for a very long time. So it's important to interview people. You'll be on the committee. It is a little bit of a time commitment, but you'll actually see some of those people will become PhD students in your program, and they'll contact you. They're like, oh yeah, can we rotate through your lab? And that kind of gets your name out there some more. I think that's all I have for now. I'm happy to talk about other things. And the other thing is obviously money management, very important. So you have your startup, so use that judiciously. Start writing grants very early. And the more shots you take on goal, the more likely one of them will land. I've gotten a lot more rejections than acceptances, but use your time wisely and use your money wisely. That's something that I feel like we don't get any training for. But think about that. And the other thing that I would encourage you to learn as far as your institution goes, there's a hiring process. There's also, if you want to let go of somebody, and so just learn about it. And it's very valuable to learn all those things. I'll be quiet now.
Thank you, Dr. John and Dr. Kim. Dr. Kim, do you want to add something else in addition to what
Oh yeah, I
we
mean, in
discussed?
terms of the like graduate student, I think graduate student matters a lot, especially to new PI, because usually graduate program acceptance rate here is like probably less than 10%, means that they are very smart. And at least they should stay here at least, 4 or 5 years to get their degree. And also like relatively their salary I need to pay is much cheaper than, postdoc level. And in terms of that, what also I did is I just tried to join different program as a affiliated faculties. That means that I can approach to different, graduate program. And also I was a search committee. That means that, so one of the senior PI just suggested that, okay, when you have interview, like the PhD program candidate, do not judge them, like you should, be nice and then try to recruit them to your lab. And ends up, I just joined as a search committee last year, and then I have a the rotation student this year. So I think it's working. So yeah, being a affiliated faculties in multiple programs in your institution, plus if you are fine with it, I highly recommend you to be a search committee in the program. So that probably makes, increase the chance to get the graduate student to your lab.
One other thing to add, if you can start reviewing some grants, okay, whether it's like through societies or not, the NIH actually has an early career reviewer program before you get an NIH grant. It just, the first time you sit on a review committee, it's just like a total eye opener in terms of what people look for, the reviewers, and it's not always the science. And so I would say I would encourage you to look into that. It's just called the NIH Early Career Reviewer Program, and they kind of put your name on a list, and like they can only take a certain number of people per session, but these are for people who don't have like an R01 or any kind of NIH funding at that time.
Do you have any recommendation like before somebody joins or takes this in becoming an independent PI position? So what are your recommendations like about having a grant beforehand versus like after you join somewhere, then you start writing grant, or you may have your idea, but you haven't like actually gotten any grant yet. So how does that go?
Do you have any comment, Taha?
Money's always good. So I would say, I think it can go both ways. I mean, always write grants. I think it's a good thing. Before there are some limitations on that though. So realistically, like if you're a postdoc or an instructor, there are certain limitations on what kind of grants you can apply for before you get that position of like assistant professor at X location, right? And so that does put some limitations on what you can do and what you can't do. I think it certainly helps. Like if you come to a department chair and you're trying to sign your contract and like you already have some money in the bank or you're bringing some money with you, it certainly helps, but it's not necessarily a requirement. It does help to, even if you've gotten some like small society grants, it demonstrates that your ideas are fundable and that you're going through and like you're writing and you're, even if it's like a $5,000 or $10,000 grant, it still demonstrates that you know the process and you've practiced it. So I do think it's important.
Yeah, I do agree, but don't worry, even though you don't have any grant, because I planned to go back to Korea, but I need a two photon system, and then the research money in Korea is not that level, and then I changed my mind to stay here. That means that I haven't applied any, NIH grant. Like a lot of people, who just, I competed with, they have K99 or, different types of research with R21, but it's totally fine. When I got this position, I didn't have any grant. However, at least I showed I tried. It doesn't need to be, fund up. Of course, if you get money, that's good. But even though if it's not, I would say it's still worth it, because writing grant, I'm still on that process. But I feel like when I write again and again and again, it's a better shape. And of course, it's so different. I submitted, but didn't get, but still, working on, versus oh, I haven't tried at all. So it's so different. I think, of course, grant, you should keep trying, but like still it's hard for me, like how can I make balance, because time is pretty limited, and then like so busy. So you need to set up your lab and then train your people. Still, you can. So I think you should make some time for grant, even though it's like a little time initially. But I recommend you to have like those time portion, in your daily life.
There is
Like,
a
yeah.
piggyback question regarding this. Does the university provide funds to start your lab in addition to any foundation grant or NIH grant in case you don't have that?
Oh yeah, I think most of like all the like school institution provide the startup money, right? Yeah, that's the like I mean probably different part because you negotiate your startup money like before sign the offer letter. But it's like totally different story. But if you
Yeah.
are interested, I'm more than happy to discuss later. Just connect like let me know, like email me.
Yeah, that's to add to that, that's a different sort of topic of like finding a job and negotiating a startup package. But yes, in general, you got to have a startup package, otherwise you can't do anything. And so it's one of those things. It's like starting a small business or like a small startup. That's kind of the way I think about the lab.
There is another question. When starting your own lab, should you prioritize getting a postdoc or a graduate student first, or is hiring a technician or the research staff? How to start set up the lab? What's the good idea?
I think it depends on what you have access to. I think it's good to get a research assistant. My minimum is usually 2 years if you're getting a research assistant, because it does require quite a bit of time to actually train somebody. But having started your lab, you need somebody to place orders and like do these other things, so they don't need to be like a super highly skilled person. But it just kind of depends what comes your way. And the other thing is, I would actually look at the finances also. So for example, at UCSF, the cost of hiring a research assistant like level 1 is very, very similar to the cost of hiring a postdoc, because of the way the union and the benefits and these things work. And so you're kind of like, okay, do I want somebody with a PhD who knows how to do a lot of stuff, or somebody who's straight out of undergrad who may or may not know how to use a pipette? And so it kind of depends on your situation. I wouldn't rush to hire people. I would do your homework, as Jinkyung mentioned, call people, and that's important.
Thank you. There is another question from Maggie, probably. Dr. Mutton, please go ahead.
Thanks. Hi, Taha. Hi, Jinkyung. So realistically, how many months before you got what you would consider up and running? Because I'm doing orders now and I'm getting estimates for like 2 months from now for equipment to get delivered. And I'm just wondering, like, am I projected to be like really going in January, or can I expect to like do things a little bit sooner? Thanks.
I think it's very depends. Like, for example, in my case, my first day was like September 1st, and then my lab up and running next January. That means that I took 4 months because I ordered most of the stuff before I move in. And yeah, I think like scheduling matters, like because it's, for example, this is probably one example, but my microscope need optic table, right? So optic table is like several tons. I need to arrange the people to move in, and also laser come in, and then scope come in, and then scope setup, and then laser setup. All those things need to, I mean, actually every steps only require hourly work or day or 2 days work. However, scheduling matters a lot. If you schedule that like very packed within a week, you know, like in advance, probably half year advance, your main equipment set it up within a week, right? And then you are good to go. But like I think it depends. But if your lab require, a lot of like molecular work based, things, like if it's in stock, it's gonna be delivered within that week, right? So I think that matters. Like Taha mentioned, probably one of the like research assistant can help you to order tiny stuff, because my first month is kind of like from morning to night, it's just order, open the box, open the box, open the box. Like, it depends, I think. Yeah. But in my case, like 4 months, but people usually between 6 months to a year, I would say. Yeah.
Yeah, I think it does take at least 6 months in my experience. So when I started my lab at UCSF, it was also the pandemic, and so you could only have, X number of people around and whatnot. But I would also, depends on like what you want to do and like what you mean by like up and running, right? Like, is the mouse protocol up and running? Do you have cell culture work going? Do you have, but I think it is nice to get something going, like that first PCR, or that first histology you do in the lab of like, just staining something that really works. You're like, woohoo, I've got like, I'm collecting, I can collect data now. And so I think that just boosts confidence a little bit. So, it doesn't necessarily mean that you're up and running and you have your big piece of equipment like going. It depends on obviously the type of science also that you do. So don't be discouraged. Like if you're around now and you're like, okay, I've been sitting here for 2 months, like nothing, like we're not collecting any data. That's totally normal. It's fine. And
Oh,
it's if you have downtime in those parts, there is no real downtime, but it would be a good idea, either write, whether it's, manuscript that you're working on from your postdoc or a grant that you're eyeing or something like that while you're waiting for these other things like the biosafety protocol and the chemicals and the somebody has to move the gas line because or the vacuum line because your incubator needs to go here and not there because it's a different size and you got a discount on whatever these things. The other thing I forgot to mention is every institution usually has like a preferred vendor and it's different at different places. Sometimes they'll give you significant discounts, particularly as a new PI. So just keep that in mind. So I know like at UCSF it was VWR, but here it's like Fisher, and like they'll give you like free shipping or whatnot. So just figure out who that preferred vendor is because it just makes life a little bit easier in terms of, if you buy your -80 from them versus like the other one, they have like a person that comes around like every week and does repairs and things. So that just kind of makes your life a little bit easier, like a year or two down the road.
Cool.
And then
Thanks so much.
probably Maggie, you are still, I mean, you are starting your lab, looks like, but
Yeah,
for
it's the boxes.
somebody cool, somebody who just, like plan to move, probably it's really helpful to have all details like photo, even, one screw in your postdoc lab. If you want to find what that specific is, that takes your time. So basically have all the labels, like, take photo, every material, and even single tool, like that save your time a lot because there's so many different things and how it look like, but if you don't have exact like, number, finding what is the right one for one by one, that takes a lot of time. And then always, if like Taha said that, okay, let's say like we negotiate something with Fisher, then they provide a like some discount rate, like discount price. But if you ask more, they sometimes do more. And then plus, like I have a lot of surgical microscope, and there is some like demonstration version. So basically, they have like demonstrate their scope in several labs, and then I mean, to me, its condition is pretty good. And then they just guarantee their quality and it's working great. But the price wise is a half price. So you can also consider the like, some demonstrated like, equipment or something. You can save your money too.
Additionally, there may be differences when you are starting your lab at a primarily research-focused institution versus when you are starting your lab at a primarily like undergraduate institution. So timelines and logistics could be different. So
I had a question kind of building off of this relating to the initial financial startup package that you end up getting from the university. Because obviously you have different constraints based on grant money that you spend on permanent, large, expensive equipment versus startup money. And there's the added level of uncertainty for potential future grant funding based on political stuff for other reasons. So basically, I'm wondering how you are approaching the purchasing of large equipment from your startup fund. I mean, particularly for things like that may be expensive. I know Taha does like single cell RNA-seq and other things. Are you strategically trying to use core materials first and find buying consumables, or are you right out of the gate spending large chunks of your startup money on permanent equipment like microscopes and things like this, as opposed to saving it for a rainy day.
Jinkyung, this is all you, the 2-photon microscope.
Yeah, I definitely start from the like main equipment I need. And then my strategy was that I check because like Ta said, equipment are the same. For example, laser, probably spectrophysics and what is it called, like the coherent, both are great. But like every different institution has a different, major vendor. That means that vendor person show up every day in that institution, and that's beneficial. Like when you have some issues or some minor health, and then figure out that, and then also I just ask quote from multiple vendor, which I know they are good. And another thing I did, like I found on faculty at WashU, she also planned to buy a 2-photon laser. That we just work together, and then we say we are gonna buy two, and then you provide better price, and then that worked pretty well. I think we got a lot of discount from there. But like I think the moment you just like ask your startup money before signing your offer letter. So I usually put the the quote price, the full quote price to there, and then once you get the money after signing your offer letter, from that moment you try to make it get more discount. It's like huge difference, I think. And then there's a lot of strategy, but it's probably a different topic. But like I would say, yeah, of course, find your major one first, and then probably the minor tools or materials later, I would say. Yeah.
Yeah, just to add to that, I think it depends on your science, right? So if there's like a piece of equipment that hinges on all of your experiments and whatnot and expertise, you should negotiate that for that large piece of equipment like ahead of time. And that item, that single item, should really be outside of your package. Like, we're going to give you X amount of dollars for hiring and, buying the PCR machine and these things, and then we're going to give you X amount of dollars for X very large piece of equipment that without it you cannot do your science. Now, a lot of institutions will obviously have core facilities, but again, it depends on your setup and what you're doing. So like a lot of the stuff that we do with like the single cell RNA sequencing, yes, we got funds for, okay, I'm going to run like X number of 10x runs or whatnot, but we're not going to have our own 10x machine, right? We can just use the core facility and it's much cheaper, right? Likewise, we just acquired my lab and a new lab. Megan Wood just started at Vanderbilt in January. We co-acquired a confocal microscope, so I didn't have my own until very recently. When she came on, we kind of share. We're on the same floor, kind of thing. Because when you start out, you don't want to spend like $500,000 on a piece of equipment that you're going to use maybe 6 hours a week, right? So it's better to take advantage of the core facilities, but at some point you may need your own, name your favorite instrument, and that's okay. So you do want to conserve your startup dollars as much as possible. Your startup dollars are actually worth more than an NIH dollar, right? Because they're unrestricted, meaning that you can spend it on, whatever you need for your operation, versus if you create an NIH budget, you have to, those are taxpayer dollars, right? So you have to follow what you said you were going to buy. There's some flexibility in it. Like if it's for personnel, maybe you said you would hire a postdoc, but you actually got a research assistant still doing the same functions or whatnot, or different, or a graduate student or whatnot. So you have some flexibility there, but those are different. Like if you buy capital equipment through NIH dollars, it's very different than when you buy it with your startup. The other thing to consider, most of us in this audience probably go to the NIDCD. The NIDCD doesn't actually have like large capital equipment grants, like for example, the Institute of Medicine does. Like you can't buy a confocal using NIH money unless you're writing like an S10 grant with like several investigators where you demonstrate that it helps multiple investigators. And so you have to, think about that as well as you're both negotiating, but also using those precious startup dollars very judiciously. So if you can, offset some of that, that'll help. The other thing to remember also is that, at the NIH, you have direct and indirect costs, and there's a bunch of things floating around about it now. But when you get money for capital equipment from the NIH, so anything more than $5,000, the institution does not collect indirect funds for that. So if you decide to reallocate that to something else in the future, it'll actually reduce the amount of money that you have, because the institution will now all of a sudden say, oh yeah, you got $10,000 for this piece of equipment and we didn't get any indirect, and you decided not to buy that piece of equipment, so we want our $2,500 or whatever from it. And now all of a sudden you're left with $7,500. And so you have to kind of think about some of these smaller things. If at the beginning, I would say the first couple of years, if you can share stuff, if you can, use core facilities, do that, and then you will need to get your own stuff eventually.
Yeah, I do agree. And then like Taha said, probably the startup money, like at least, big equipment wise, that's probably almost the only chance for you next 5 to 10 years, have your own equipment. Because a lot of like institution like said that, oh, you can use, core to photon, but I just, keep asking that, like, I do need my special, like animal setup with the speaker and then, hearing physiology. I do need my own two photon scope, because I learned that it's really, really, really hard to buy your equipment with NIH grant. And then even though like there's some equipment grant, but like Taha said, it needs like not only for you, just multiple PIs. So probably startup is the like really great, important moment you guarantee your own equipment, main equipment. So just keep in mind, and if you really need it, you should, like keep asking when you negotiate your position.
Yeah, the other thing, the other strategy, like depending on the institution you're at, like sometimes you need a piece of equipment, like Jinkyung mentioned, like maybe it's demo, but maybe the lab next door, the person is retiring or they're moving or whatever, and there's like a bunch of equipment they're giving away or whatnot. There are certain things that you can take a little bit of shortcut on, like for example, that dissection microscope maybe needs like a new optical thing that costs $3,000 as opposed to spending like $30,000 on the thing. And that's fine. But there are other stuff that's like a little bit more sensitive. Maybe you need that PCR machine that's the high grade one for your single cell RNA-seq experiments. I probably wouldn't take shortcuts with that one, right? Because that has a lot of things hinging on it, and you don't want to, so be a little smart about that. But if the lab next door or down the hall or whatnot has some surplus, like test tube holders, whatnot, or those things are expensive, like go grab them, they're free. And so it just depends on like how you can be very judicious with both your startup and your NIH dollars. There was another question in the chat.
Yeah. What have you done to make your lab and research distinct, probably diverse from your previous postdoc group? And for example, how does this factor into applying for grants?
Yeah, I think one of the things that's important is that becomes a lot harder to do if you're at the same institution as your PI, right? So when you're a little bit further away at a different institution, they can't just claim, oh yeah, this is just your this is your PI's data or whatnot. I think demonstrating that your lab is up and running is important when you write that first NIH grant. Like here's a histology piece that is it's not new data like other people have done, but I did this in my lab, like I did this, I imaged it and whatnot. So those kind of like little things matter. And then obviously your idea is important. You don't want to be using the same ideas as your PI, but it's also important to actually chat with your PI if you're on good terms. Hopefully everybody's on their good terms with their former postdoc PI, and they'll give you some advice on how to do that. They're like, oh yeah, like we're not planning to write a grant on like this X number of direction of whatnot, and you can be transparent about that in your grant writing.
I can see the question from George. He's asking that like getting postdoc or graduate student first, or like technician or research assistant. Graduate student maybe like you may need some time to get first one, but like if you ask hiring postdoc or technician, it's I think depends what level of help you want to get. But in my case, I think like simple ordering stuff I can do. Then usually, typically people hire one technician and one postdoc. But I hired two postdoc instead of one technician, because I think I can do like, some simple order work at night, and then I want more people to perform research. So, but I heard that like a lot of my colleagues got a lot of help from technician when they start lab because there's multiple things to order and that's a lot of work. So I think it depends. Yeah, I think Dorsey, you have a question?
Yeah,
Please
thanks,
go ahead.
and Jinkyung. I guess so. I just started, and so I'm like trying to set up my lab. So this is great timing. So in terms of like the animal protocol, would you write like all potential strains of mice you may or may not use in the future, like when you're trying to like plan it, or would you like just submit it and like add strains later if
Uh,
you think
My advice would be just submit it with the minimal number of things to get it approved. So wild type crosses, breeding, we'll talk about experiment because it's a really a long process to go through it at every institution I've been at. So I would get yourself a protocol number, and then I would submit the amendments and let them roll through. Obviously, you don't want to break any IACUC rules, but I would try to get a protocol like approved as soon as possible with
Yeah,
a
I
minimum number
also-
of bells and whistles.
Yeah, I also did it for probably next year first, and then I'm adding. And also it depends, like Amanda, like at WashU is pretty, I feel like it's quicker than Stanford. Stanford took about like 2 months, but here is like 10 days or about like something like that. So adding something is not easy. It's not hard, but like probably initial revision, maybe you may go like multiple cycles and take some time. So like Taha said, make it simple and then get approved, and then you add more and more and more.
Yeah, it totally depends on where you are. So when I was at UCSF, my lab was at a site that had an animal facility, but they had this rule that the animals cannot leave the animal facility. Like they can't come up to your lab. And so it took 7 months for me to get that approved, where they would allow me to take the mouse out of the mouse house to the lab. We still did it, but it took 7 months. So I think 10 days is very unique. That's awesome. That certainly doesn't happen here at Vanderbilt. Usually it takes on the order of like 4 to 6 weeks, I would say. And usually what happens is some like administrative person will review it and be like, oh yeah, like you forgot to put a period over here or whatnot. And then it bounces back to you 10 days later and whatnot. So just I would try to, again, not put as many strains and not as many like animal surgery and things like that. I would try to keep it at a bare minimum until you get that first protocol, then submit an amendment.
Cool.
And then having people like, who helps, related to animal transfer or animal order, and then like, all the dead individuals, and then kind of be nice and appreciate their support and then become their friends. That actually makes the like process much quicker. That's my like experience. Like, how can I explain? Sometimes they just prioritize my request because, okay, I do need this, for next, proposal or something. Could you help? Like, if they know you, they try to help. So, like, having people around you, of course, chair or your senior colleagues, you're gonna be really, have a great relationship with them. But don't forget about other, a lot of people's in the department, and then, a lot of different sort of in the institution, there is a different level of people. And they always know who they are and then appreciate their support and then become their friends. I think that helped a lot.
I'm just curious, other than animal protocol, if somebody is planning to use like human iPSC line or human stem cell lines that have got purchased from vendor, so is it like still needed to get IRB approval for those cell lines before you use, or is there some other way?
I don't know the rules about that specifically. I mean, we use human tissue, but it's from the operating room. We need an IRB for that, but we don't use any cell lines like IPS cell lines specifically. So I would look into the institution's rules. And then the other thing is if anybody's doing any sort of stem cell work, I would actually check what the rules are, because that landscape is quickly changing at the NIH, particularly if you're working with like embryonic tissue and whatnot. And the rules are different, for example, at the federal level versus like in California. So I differ on that one.
Okay. Another question I had about was like, how do you find your work-life balance, like particularly when you are leading the lab, and if you for Dr. Tarzan, like he's a clinician as well, so how do you manage your work-life balance?
I don't have work-life balance at this moment, so I may not provide the right answer. I don't know, this is really hard, but usually daytime, I don't have any time to write something, so I need to write something night and during weekend. And yeah, so Taha, do you have better answer?
Yeah, I mean, there's no answer. I think it's different for everybody. I have 3 young kids now, a 6-year-old, a 3-year-old, and a 7-week-old at home, and my wife's
Oh my
also
God.
full-time working. She's a physician, and so it's always a struggle, right? Whether it's, when you're starting out first, you got to check on your animals, make sure your patients are okay in the clinic and operating room, and make sure your kids are fed and like, alive also. So it's always a balance. But one thing that I've learned to do is protect my time while I'm at work and try to be more efficient. So for example, I know that I'm not as efficient at like writing and thinking in the afternoons. So if I have any grant writing or paper writing, or if I'm sitting down to do some data analysis myself, I will block that part on my schedule, like, 8:30 a.m. to noon. It's blocked on my schedule. I try not to put meetings there. The other thing is my clinical admin person has access to my schedule. So I always put like lab there. So they have to email me if they want a slot there. So it used to be when I would start, they're like, oh yeah, we put this 10 o'clock meeting for like, whatever, like either somebody's coming to interview or you have some committee thing or whatnot. So I was like, no, like I cannot do mornings. Like it has to be in the afternoon. And so I've learned that by myself. It is always a juggle. Like when I'm at home, I try to make it home for dinner so I can have dinner with my kids and my family and like put the kids to bed, because you get like a short amount of time to spend with them. So I try to do that, and then afterwards I got to have a little bit energy left to maybe reply to some emails or whatnot. And I also try to do some work early in the morning if I can. It's either between 8:30, 9 p.m. to like 11, or early in the morning between 5 to 7 a.m. To try to get some of those things done. The other thing I like to do is throughout the day, I like to keep the tasks moving. So there's a lot of things that come your way that's like a quick, like yes, no, or like a one-liner versus sitting down and like doing something that requires a little bit more work on your end. So it's important to kind of keep the tasks of the day moving as you're going along, so that you don't have like 50 small tasks by the time you have that 1 hour block or 2 hour block where you can sit down and think or whatnot. So that's just one other strategy that I use. And then on the weekends, sometimes I have to go in and do an experiment. I've been fortunate. We have good people in the lab now. You try to get to that point where there are certain skills that you have, but you want to pass it on to somebody else, so that when the next person joins the lab, you can tell them, oh yeah, go ask so-and-so. They will teach you how to dissect the inner ear, because they will go check on the animals. And so you have to do a lot of that stuff at the beginning yourself, right? Like you have to teach them how to dissect the inner ear, and you have to take them through the protocol for immunohistochemistry and learning to using the confocal, or whatever it is that you're doing, whatever setup. But there are still some things that you still have to do. But the more you offload and delegate to other people, even if they make mistakes, it's okay. As long as you've got good quality control and you meet with them, you want to be able to offload some of those things so you can take on some of these other tasks, whether it's you getting, going to your dental cleaning or dropping your kid off or whatnot. But it's always a struggle.
Thank you.
Thank you, Taha. This is really helpful.
Agudama wants to ask a question.
I have a question.
Please go ahead.
Yeah, so just started 3 weeks ago at University of Utah. My name is Agudomo, and yeah, I think time management has been really stretched to the extreme for me. And I wanted to ask that question actually, but on a separate topic. So I'm wondering how do you schedule this grant writing, sort of like uh, sort of like daily habit. How do you just consistently write for the whole year, or do you take a break? And I think the more you write, the more you submit grants, the better chances of getting grant, right? And but I also heard I also get recommendations or suggestions of not submitting too many grants because you don't want to have like a bad reputations of constantly submitting grants to NIH either. So how do you sort of like schedule your grant writing?
I think so. I think you can't write the grant like for a year, right? Like I think like having a deadline is good. And then I try to write something, every 3 months. I mean, like maybe this time R1 and next cycle maybe R21. But like I wouldn't resubmit the same version to same like, the same like round or same NIH, study section. That may not be helpful. But I think when you try more and more and more, it's improved. But you need to edit, right? And then you can do like you can like apply for all the grant, right? Your time and energy is very limited. So like pick the grant proposal you think your research field is like greater fit than the others, and then have some deadline. It depends like what do you want, but like in my case I try to have like every 3 months, but sometimes it's foundation, sometimes it's like NIH grant, but try to keep applying. But of course some research part is overlapped, but like of course it's not shouldn't be the exactly same. So that's my strategy.
Yeah, one thing I would recommend. So first of all, so the NIH has new rules now, so you can only apply six grants per calendar year, and that includes your R01 submissions, R21s, any resubmissions as well, or anything where you're a co-PI on. So that includes multi-PI grants. So that's one thing to consider. That rule just comes into effect actually September 25th of this year, and so total of six. The advice that I got, which I followed, was apply for society grants right at the beginning, and then also apply for NIH. I don't think there's, the apply. I don't think there's as many things as applying to too many grants, but you still want to be picky and choosy, because the nice thing about the grant is even the grant that gets rejected, you get some feedback. So it's important to take that feedback seriously, not just throw something else out there and just say, well, I'm just going to keep taking shots on goal, and it's moving. If you play soccer, it's moving like not even like close to goal, right? So but you want to hone that in. I think that feedback is important also from the scientific community, from your peers, right? Maybe more senior colleagues and whatnot. So the advice that I got when I came to Vanderbilt was to apply every cycle, which is kind of aggressive. Like my department chair, who's a clinician scientist, it's a little bit different when you're just starting out than when you're somebody who has multiple senior people in your lab who can say, oh yeah, prepare this figure for me, right? Like you can't do that. You have to do it. And so I would be judicious about the first few, but try to get things on paper, try to collect prelim data. I think all of these things will help, but it is a balance of time, right? When are you going to collect data? When are you going to hire that person? When are you going to, write that first paper, kind of thing? So all of these things sort of add up. So don't feel like you have to apply every, like every cycle the first, 2 or 3 years, but you do need to apply some, right? And the more it's just like another skill. The more you practice it, the better you will get, and the more feedback you will get. I don't know if that answered your question or not.
Yeah, absolutely. Thank you.
I mean, getting feedback before your submission from your, senior colleagues, that would be really helpful. Like
Right.
at least, I mean, in my case, I realized that, oh my God, I need to rewrite everything, like, because like I really haven't seriously, applied like R1 as a independent PI, right? And that helped a lot. So getting feedback from your colleagues, that's gonna be really, really helpful, I think.
Yeah, and to go along with that, your colleague doesn't need to be like an inner ear biologist if that's what you're doing. In fact, sometimes it's helpful to get some feedback from somebody else who's a well-funded R1 investigator. They look at this and they're like, yeah, this is not clear, like clarify this. And that's actually been super helpful for me applying for we recently applied for some R21, and that was very helpful from my department chair, who is a head and neck surgeon. He does like cancer biology. He was like, this stuff doesn't make sense. I was like, oh, okay. I rephrased it, and it was very helpful feedback.
Yeah, recently I applied. Oh, go ahead. Sorry.
No, yeah. The other thing I was going to say is maybe you don't have a critical number of auditory neuroscientists at your institution, and that's okay. You can still get feedback from very smart, well-funded people that are your neighbors or whatnot.
Yeah, recently when I applied the foundation grant to neuroscience field, mostly they are brain people, and then I got a comment from just who is working with the brain, not auditory periphery, but it was really helpful because none of like the people in our field couldn't recognize, oh, that brain people don't know about that, So in terms of that, getting the feedback depends on what grant proposal you are preparing. So when you think about it, that's gonna be really helpful too.
Yeah, well, I will share my specific aims right now today to all of my colleagues. I think that's going to be really helpful. Thank you.
Good luck.
Thank you.
Good luck.
Thank you, Taha and Kim. Thank you so much for sharing your valuable insights. And is there any final comment or final thoughts about or take-home message that you want to comment on before we conclude this session? We are already 2 minutes past this scheduled time.
I would just say, yeah, you are doing great, and then your lab is gonna be, running, and then with great people. So I think you are doing great now. I would say that. I mean, to myself too.
Yeah, I agree with that. And the other thing is, reach out to people. I think it's nice to have very senior mentors, but it's also nice to have people who just went through it like a year or two ago, because it's different for the big boss who's, been doing this for 30 years versus, like, oh yeah, I struggled hiring somebody, or I struggled with this grant like last year. Let me tell you the mistakes I made. And so it's nice to build that collaboration community. Like find Jinkyung and I like at ARO, come chat with us. We're happy to always grab some coffee and co-commiserate together. And that's, I found that very useful as well.
Thank you once again. And many thanks to our audience for asking these wonderful questions and for making this coffee hour interactive. And I'm sure that this audience got new perspective about how to making this journey of starting your own lab successful, and they will get benefited, particularly when they have to make such decisions while starting their own lab as a new PI. Thank you all. Have a great rest of your day. Thank you.
Thank you.
Thanks, guys. Bye-bye.
Bye.
You.